and then in the afternoon we will speak about post-COVID depression. Actually, this will be broadcasted on Thursday as the introductory plenary lecture at the CINP in Taipei. And so you can see it in advance. It's me, always, they are nearly the same slides. This will be a very long talk, but we can cut it short if it is too long. But anyway, I thought it was important to show you what is our current perspective in these terms. The literature is very huge, and we can start seeing this. Also, I'm not using the microphone, but if you want, I can use it. Usually, I don't need it. So, capitalizing on the huge work done in neuroimmunology, we know that neurons do speak in the immune system, okay? Microglia resident in the brain is essential for the synaptic homeostasis. Actually, every night while we are sleeping, microglia is doing its work on the synapses, changing them. And neurons do speak also with the immune system, and they receive messages from the immune system. Neurons and oligodendrocytes do express receptors for cytokines, and oligodendrocytes do respond to cytokines because they are the main trigger of inflammation. They can start inflammation, and if there is inflammation into the brain has to change in order to defend itself from pathogen. So, what the brain does is to make a space. Oligodendrocytes, when they receive messages, and especially TNF-alpha, interferon gamma, interleukin-8, we will see them, do demyelinate. They make more space for the water to flow. Of course, this makes connections between neurons very difficult. It slows down connectivity, and it makes what we will see this afternoon, this strange kind of brain fog which follows great infections with cytokine storms. But the point here is that even in the absence of acute infection, patients with, let's say, endogenous major depressive disorder or bipolar disorder do have raised seconds elevated biomarkers of cell-mediated inactivation and inflammation. There is a deregulation of the immune system with high peripheral markers of macrophage monocyte inflammatory activation patterns and activation of microglia into the brain. And these first introductory slides are just to tell you that this perspective is not in contradiction with the traditional perspective followed in psychiatry, which shows that patients with mood disorders have cyclical episodes of illness which follow a seasonal pattern. We know very well that across the season, here they are in Australia, so winter is in June, serotonin decreases into the brain, and it is highest in summer. Actually, in a couple of weeks, we will arrive at the summer solstice, and this will be the high serotonin level in the brain, more than 1,000 picomole per minute produced, and then it will go down 10 times during autumn, and will come up 10 times during spring, following the change in the photoperiod and the availability of daylight. And we know that depression strikes in spring and in autumn when serotonin have to change. We also know that patients with seasonal affective disorder, with the seasonal pattern of illness, do have a higher change. This is the difference in the serotonin transporter from winter to summer. And this change is proportional to the symptomatology. It's quite linear. Why this neuroinflammatory perspective is not in contradiction with this? Because the immune system goes together, and probably these changes in neurotransmitters are also part of this yet not completely known interplay between monoaminergic neurons and immune cells. Look here, this is the only study available on this topic. My friend Raymond Lamb took patients with winter depression, and you see that they have a higher macrophage activity
compared to healthy subjects. And they have a lower lymphocyte activity compared to healthy subjects. But when they are treated with light therapy, only light, without drugs, and they heal from winter depression by acting on their biological clock, they get exactly to the same levels of healthy subjects. So, acting on the biological clock in winter depression does act on the immune system, and it does also act on monoamines and on serotonin. And you can see here inflammation. This is TNF-alpha, this is interleukin-1, the core of the inflammasome, and they are higher when there is this activated innate immunity, and they go down after successful treatment. This is a recent study. We don't know very well what happens over the season to the immune system. This has been published just 1 year ago, and it is the change over the season in healthy people of, you can see here, inflammation, C-reactive protein, and lymphocytes and neutrophils, which are a good marker of innate immunity and the inflammation, because monocytes do not change so much in number. They can activate, You can find into the monocyte patterns of activation, but their number remains almost the same, and the pattern is quite clear. Now, in June, we have the lowest inflammation, the lowest C-reactive protein, and the highest lymphocytes, which were in spring. And the possibility is that these innate variability in immune parameters will influence human susceptibility to disease. We got struck by the viruses when we have the highest levels of inflammation and the lowest levels of lymphocytes in winter. And in summer, we become again able to fight every pathogen. We don't know why this happened. It has just been described 1 year ago, but this is quite a complicated paper, trying to put into relationship cells, populations with all the proteome expressed in the blood, and they show that there is a longitudinal variability over time. These people were sampled 4 times over the year. And it is an individual feature. And there are guys who are more variable in their relationships between cells, cytokines produced, and these are the people who are more sensitive to metabolic dysregulation. They got fat during the winter, and they have a lot of problems like diabetes linked to this instability in the immune system. Again, these are very new findings, and everything has to be done. We don't know very well what happens. This is a beautiful review published now by these people, showing that in the health of the woman who is especially sensitive to mood disorders, there is a continuous change from innate and adaptive immunity during her life, with the grid cycles of menstruation, pregnancy, menopause. During the pregnancy, there is a shift. Adaptive immunity, the one used by lymphocytes, goes down, because otherwise the mother would attack the fetus as an extraneous being. And then after childbirth, there is a raise in adaptive immunity. Now we know that if this raise does not happen as it should, the woman will develop postpartum depression or psychosis during the pregnancy. And these changes are explored now, okay, in this lifetime of women. Estrogens, steroids, and the peripheral immune system are closely linked, and could be again one of the ways to explain this special sensitivity of the woman. And these are the 2 levels, okay? I will not describe them, of course, in details, because otherwise we will stay here all the day speaking of immunology. But innate immunity is inflammation. There are a lot of cells. You can see here the macrophage. Of course, there are natural killer, basophil, granulocytes of the kind, and macrophages. And probably at the top of the pyramid, there are the natural killer cells who can express cytokines. And then there is adaptive immunity based on the production of antibodies around T cells that can target selectively pathogens and selective targets. This is slow, while innate immunity is very fast and violent, and we need both, okay, to survive the attack that our body suffers. Of course, if we have a fast adaptive immunity, the faster we have and the younger we are, the faster adaptive immunity will be, and the older we are, the slower adaptive immunity will be. The faster adaptive immunity will lead to a rapid clearance of pathogens, as in the case of COVID, as we will see. So we need both, okay? This is violent. I put a nail here covered in blood because innate immunity and inflammation is more typical of males. It's stronger in males than in females. Females are more able to have a rapid adaptive immunity targeting a single target without producing such a cytokine storm. And this is, by the way, the reason why women survive well to COVID infection more than males, because they have a more unstable, and so they can suffer of depression, but also rapidly reacting immune system, okay? So here we are, and there are two main parts of this talk. We will see that patients with mood disorders have altered immune inflammatory response. We will try to define a phenotype and see its effect on the brain. And then we will shift on therapy by showing that immune inflammatory mechanisms can be possible targets for psychopharmacology. Higher inflammation hampers response to antidepressants. Antidepressants share immune modulatory and anti-inflammatory properties, and anti-inflammatory and immune modulatory treatments can be used as antidepressants. I will try to stay short and give to you bullets, okay? Very short bullets about all this, because it's a huge area of research which is growing day by day. So starting with the phenotype, the problem is that it is not stable, okay? It's an unstable phenotype. I will summarize in the following slides the work we did in a European-funded consortium, which is called Mood Inflame, and then it follows by Mood Stratification, which investigates the inflammatory response system for the early diagnosis, treatment, and prevention of mood disorders. The idea is that probably we have a first hit in utero during the development of the fetus. It could be, of course, there will be a genetic vulnerability, of course, but it could be infection or stress, changes in steroid hormones of the mother, stress of the mother, all the modeling in animals tells us this. This first hit leads to a first activation of monocytes and a priming effect on microglia, which seems to become more reactive for the rest of the life of the people stressed here. And then we have this HPA axis priming and cytokines already at the level of development of the brain. This brain, once aging, needs, however, that's the theory, to be hit again by a second hit again. Stress, sex, steroids, and the postpartum status is a beautiful example of this, and possibly microbial danger infections, which can trigger this abnormal response, these huge responses of microglia, which leads to an abnormal function of nerves, and it leads to this activation of inflammation in the periphery. So we have a lot of effects, autoimmunity or infections that can trigger this. What we show in the patients with cross-sectional studies is that during the life, the phenotype changes. There are early T-cell imbalances followed by a low-grade inflammation, which persists throughout life. But first it starts with a deficit of T-cells. You can look at moodstratification.eu if you want more news about this. These slides are courtesy of Raymond Exager, who is the coordinator of these European studies. This premature monocyte and T helper cell aging starts in the young adult life and worsens over time, okay? And then it leads to this raised C-reactive protein, interleukin 6, whatever, and to abnormal function and structure of the brain during the adult life. In major depressive disorder, the process is slow and it worsens over time, but what we see is that in the presence of childhood trauma, it moves earlier in life. So traumatized children do already have these altered set points of inflammation. Soon after the trauma, they last throughout the life, and we know they have earlier onset of MDD and worse episodes over time, and even worse outcomes, because they have an upregulation of the inflammatory genes since they are very young. In the case of bipolar patients, the things are worse. Patients with bipolar disorder seem to have a T helper deficit and signs of immunosenescence already in the teenage time before the onset of illness. Then, after the first episode of illness develops, at every episode we have flares of inflammation which go together with the episodes and lead to again, aging, immunosenescence, T helper deficits in the patients. So at every episode we have a flare, okay, which activates and we think could damage the brain wave after wave of inflammation. I will show you. I think the most beautiful example of all those is the postpartum status, because we see that in postpartum depression, T-cell defects are really apparent. You don't have the rebalancing or adaptive immunity which is expected after the partum. Mothers stay with these high levels of inflammation, and high levels of inflammation correlate so strictly with depression. I didn't put these slides because there are too many already. That based on the levels of cytokines in the last weeks before childbirth, you can predict depression in the mother 1 month later, and you can even think that, because it's madness. If I had said these things 20 years ago, they would have said that I was mad. You can predict how much the son or the daughter will weep. So its emotional state is linked with this inflammatory state of the mother in the last weeks before childbirth, and we think this is because all these variables act on neurotransmission. It's a growing research. Actually, it's what I'm doing now. I'm studying postpartum depression in this exact moment. In the last paper we published, which is now, it's out now, it was accepted December 21, we saw that there are patterns of activation of genes into monocytes, okay? This is activated monocytic macrophagic immunity, activated immunity. This cluster of genes can be affected by what is here CA, which is childhood adversities, childhood trauma, or cannot be affected by them. Some of these clusters are the same, and they are activated the same in patients exposed to childhood adversity and not. Some are activated only in patients exposed to childhood adversities. And this probably explains why people with childhood trauma have a worse inflammation, because some clusters of genes are specific to them. And when you look into the macrophage, what happens, these things do influence mitochondrial health and apoptosis, inflammation, chemotaxis, coagulation and adhesion to the endothelium, and so endothelial homeostasis, and cholesterol, and the pyroptosis, which involves many cells in the body of our patients, and the production of oxidative radicals, and a lot of stuff, to summarize, which have been associated with mood disorders and never explained. We know that our patients have a higher risk of developing any kind of metabolic problem, and probably this is linked with this pattern of activation of macrophages, which you can see here is quite specific of mood disorders. These are first episode psychosis,
which then will become bipolar disorder or schizophrenia and other things. And you can see that the response to activating effects and the production of interleukins by the macrophages is not so much apparent for three stages of psychosis which are not in the course of affective disorders. So it's quite different. A marker which can be easily seen is the blood cell count ratio, because of course we can't go so much in deep in our clinic, but we can measure neutrophils to lymphocytes, monocytes, platelets, and all this kind of stuff, which are easy. By the way, there have been beautiful papers about inflammation immune disorders produced in Turkey. I will not summarize them here because I guess you know them, but I will give you my perspective, which is not the same. But these things have been known. You can see the rise in the number of papers about the neutrophil to lymphocyte ratio in the last years. Nobody cared. I mean, we had these things in our charts, but nobody cared. In the last years, you can see the number of papers in Netherlands, which are thousands now, because we have higher neutrophil to lymphocyte ratio both in bipolar and in MDD patients, as we showed here in this meta-analytical work we did. And if one can go in deep and measure cytokines. This is a paper we published, and you see blue lines are healthy controls. For some reason, I don't know who chose these colors. The red or what color is it? I can't see it. Are the MDD patients, and the gray one are the bipolar patients. And you can see that in a panel of 40 cytokines and chemokines, all these measures are higher during depression than in healthy controls. And when you try to diagnose them, you can find the signatures which can allow you to discriminate patients from control, but also the categories of patients, bipolar disorder from MDD, because the patterns of chemokines and cytokines is slightly different, but consistently different. And so we have a lot of cytokines which favor the diagnosis of MDD, a lot of cytokines which favor the diagnosis of bipolar disorders, and these are mostly chemokines and chemoattractants for macrophages, okay? Because the T helper and the adaptive immunity deficits is worse. This is all new, I'm telling you. I think it's the future. I think in the future we will drink just a small amount of blood from a finger of our patients, and we will say, okay, you are highly inflamed, and so it's most probable that you have to take lithium in order to not develop bipolar disorder or things like this. But we are at the beginning, okay? If we go into the CSF, into the CSF, we find similar things, but there are very few papers about this. Here you can see that it is completely different, the pattern of schizophrenia from bipolar disorder and depression. And what is activated in depression is interleukin-1 beta, kynurenine, which we will see, and kynurenine acid, TNF, interleukin-8, and consistent. These are, I don't know why it changes. I don't know what happened. I think I pressed the wrong button when I was preparing the slides, but anyway, it's nice to see. This is our acute phase response signaling. This is the incredible thing. So we find signals in the CSF of our patients in the absence of apparent infections, which are suggestive of acute phase reaction. Something is going on in the brain. We don't know what, but something big is going on. And I think we should study this. Okay? And this activation of acute phase response has been linked mainly with interleukin 6 and the activation of STAT3. But these are all papers coming out in these years. And so we just can't say
fact, what we find in the periphery is measured in the CSF of the patients by high activation of inflammatory markers. And why does this happen? We don't know, but I am anticipating a slide which we will see in a few hours, because Robert Nurken is the guru of this. He clearly showed that association of seropositivity for influenza and coronavirus is associated with history of mood disorder and suicide attempts. And more recently, in this paper, which has been published a couple of years ago, that patients with MDD have altered patterns of reactions to viruses, in particular Epstein-Barr viruses, but we can think everything, okay? So it could be that the immune system of our patients does not react in normal ways to pathogens, and this could be one of the triggers of this long-lasting inflammation. We don't know. These are papers, last Congress I was in presence was the American College of Neuropsychopharmacology in 2019, when these data were presented, and everybody was speaking about this. Then it came the COVID, and it was an incredible experiment, proving a lot of this stuff, but also changing the world, so we don't know. This kind of research stopped there, okay? And we are at this point. Does this have an effect on the brain? A lot. Really a lot. This is a famous review of some 13 years ago. We have that the peripheral inflammatory response matches activation of microglia into the brain. Somebody thinks that monocytes do cross the blood-brain barrier and enrich the cells into the brain, but this is difficult to prove, of course, because we should do biopsies, and this is not so good. But anyway, either the resident microglia or cells coming from the periphery are activated. They release cytokines, chemokines, inflammatory mediators, and astrocytes do respond immediately to them and decrease the glutamate reuptake. So in the synapse, we will have glutamate spillover and excitotoxicity. On the other way, the oligodendrocytes are extremely sensitive to all this. They will lack the trophic support by the astrocytes and directly respond to the chemokines, and either they myelinated or kill themselves. They go toward apoptosis in case of high inflammation. Of course, oligodendrocytes can regenerate, and so the idea is after the problem is solved, the oligodendrocytes will regenerate again. But you know, it's a big trouble. And when things go really bad and people kill themselves, either if they kill themselves because they are affected by affective disorders or by a depressive episode in course of schizophrenia, for diagnosis of schizophrenia, they have activated microglia post-mortem throughout the brain, in all the areas, and we will see with a special concentration in the atrial cingulate cortex. We can measure the activation of microglia in vivo, but we don't do it because of money, of course, because it's costs, and there are a lot of limitations, especially here in Italy, to the use of radioligands. But there is a beautiful radioligand for the translocator protein, TSPO, which marks activated microglia. As you can see here, they started to use it in neurology, in the neurological disorders such as multiple sclerosis and all the conditions with inflammation, and then it is started in the research in psychiatry. It is expressed on the mitochondria of the patients into the microglia. When microglia activate and changes from giving support to the neuron to preparing for battle and releasing cytokines, this translocator protein is highly expressed and probably is part of an anti-inflammatory mechanism by which microglia self-inhibits itself. Anyway, we can mark it with several markers. A very famous one is PK, you will see it, and with positive emission tomography we will mention. And you can see here marked cells
do overlap with PK 11195, I will call it PK from now on, okay? This mark, it's easier. With PK, they overlap with staining for activated microglia. It's exactly marking. There was this pivotal paper published 7 years ago by Meyer and workers in the second world, and they showed that throughout the brain, in all the regions you can see, patients with a major depressive episode have higher levels of activated microglia than healthy controls. And this binding correlates with severity of depression. That was the first paper about those, mostly cited, and it tells us that during a major depressive episode, microglia is activated and severity of the picture links with it. And look here, patients with suicidal thoughts are the worst activation of microglia. This matches the post-mortem finding of activated microglia in the brains of suicides. Here you have late life depression, when we have even probably higher levels of inflammation, because there is immunosenescence, which is also paraphysiological. Everybody goes on and the immune system goes on, and these guys, when they are depressed, have a higher level of activation of microglia, which concentrates, look here, in the anterior cingulate cortex, in the perigenual anterior cingulate cortex, which has so much been studied in mood disorders and associated with abnormal structure and function, as we will see this afternoon. And here, very interestingly, you see again that cognitive dysfunctions in patients are linked with activation of microglia. Of course, you have inflammation in the brain. It's like you are having a fever always, and the people with mood disorders who have a lot of problems in their condition do have them related to activated microglia. And so psychomotor speed, attention, cognition is associated with this activation. This is very important, I think, and it is a product of these mood inflame studies that I mentioned, because these are euthymic patients. These are bipolar euthymic patients. And the difference in respect to controls, it is not so striking, but there is, and it is significant. So we can say that activation of microglia do persist into leukemia. These patients had been stable for at least 6 months and were not taking drugs, okay? And they have signs of activation in brain. And you can see in a second study that this associates with N-acetyl aspartate, which is a marker of neuronal well-being. And the highest activation of microglia, the worse the well-being of neurons in leukemia. So what those triggers, we don't know. I have no answer. But this is what we know that happens when there are encephalitis or autoimmune diseases or inflammatory diseases which target the brain. First, microglia activates in terms of hours after tissue damage, infection, whatever, a stroke, let's say. It activates, and then there is a long-lasting activation which leads to localized inflammatory responses, microgliosis, astrogliosis, and then cells are called into the brain. Monocytes, lymphocytes are called into the brain, and they will stay there. And then there is the resolution of inflammation and the return to the normal condition when it is possible. There are few data about the infiltration of cells into the brain of the patients, but these are very good data published again just before the COVID, during the COVID actually, in the brains of the Magdeburg University. These are the post-mortem brain collections of the previous East Germany psychiatric services, and they show that there are
into the brains of patients with mood disorder and also schizophrenia, higher levels of infiltration. You can see here one patient with mood disorder, 37-year-old woman died, and you can see here marked cells, which are T cells, throughout the brain. Look here, it's incredibly terrifying, I have to say, because these things do happen when there are big problems in the brain, and nobody looks for them, okay? And you can see here, in the case of acute schizophrenia, there's not so much, but in residual schizophrenia, where the brain is highly damaged, there is a high level of infiltration by T helper, which is present in mood disorder throughout the lifetime, okay? While there are not so many B cells, but a lot of T cells, as if they were looking for something.
I told you, this has consequences on brain health. A first thing is that it wipes out several neurotransmitters. Activation of microglia will lead to the production of a lot of cytokines which activate the indolamine deoxygenase, which breaks tryptophan and produces uranic acid and quinolinic acid. This means that tryptophan doesn't go to serotonin, into the brain, but those go to kynurenine. And this kynurenine to tryptophan ratio is one marker that can be measured also in the periphery and exactly matches what happens into the brain, because macrophages are activated also in the periphery. So you can see both. No, you don't need to go into the brain. So we have this activation of indolamine deoxygenase, and then there is kynurenine, which can go to quinurenic acid, which is an inhibitor of NMDA, and so an antidepressant possibly, like ketamine. Or to quinolinic acid, which is an activator of NMDA, and so in animal models we see a sickness behavior when we activate IBO followed by a depression-like behavior, which is due both probably to the decrease in serotonin and to the activation of NMDA, which go together into the brain of our patients due to their inflammation. No, possibly, I don't know because nobody studied it, but when we give ketamine, possibly we antagonize this mechanism which is linked with nerve inflammation, stimulating the NMDA, which is abnormal. And when we study this quinurenic acid, this was one of the first papers about this, we see that we can define a tryptophan breakdown index, quinurenic on tryptophan, and a neuroprotective index, which is quinurenic acid on quinurenic, so quinolinic acid. And we see that the breakdown index is higher in patients, and it goes down to quinolinic acid, because in case of inflammation, this is the preferential pattern. In my group, first this one before, this is the level of quinolinic acid in the CSF in patients who survive suicide. And you can see that months after suicide attempts, there is a persistent inflammation, and quinolinic acid is lower. So it is a matter of severity and of bad outcomes. And we associated it in my group with both phases of the illness. These are healthy controls, tryptophan. This is depression, and this is mania, because also mania is associated with immunoinflammatory activation, and with white matter microstructure throughout the brain. If we know this leads to white matter, we know we have to run. Patients over time, at every episode of depression, do decrease their memory. They have a progressive decrease in cognitive abilities, which worsens every time. We associated this with a change in white matter microstructure, with a reduction of fractional anisotropy and an increase in radial and limb diffusivity, which suggests disruption of myelination throughout the brain, psychomotor coordination, executive functions, working memory, attention, and information processing as associated with this reduced fractional anisotropy, increased limb diffusivity.
So, with more water flowing into the brain and a lower directionality of fibers and congruency of the fiber tracts in limiting structures throughout the brain. And again, you are with the same panel of cytokines which we had associated with the diagnosis of mood disorders in our samples, which associate with cognitive functions, okay? Reducing them. Higher cytokines in the periphery, lower integrity of white matter in the brain, lower cognitive functions in the behavior of our patients. And this also marks a bad evolution, a poor antidepressant response. It's difficult to read because it's red or black, but it's written here that the degree of improvement in terms of changes in the Hamilton scores negatively correlates with MB and RD. So the worse the white matter structure, the worse the improvement with antidepressant treatment. And this, I think it's one of the findings I'm most proud of. We associated these changes with cytokines. Interferon gamma, TNF, interleukin-8. This is the reduction of functional anisotropy, the increase in mean diffusivity, the increase in radial diffusivity because oligodendrocyte dymelinate, and we showed that the higher the peripheral levels of these cytokines, the worse the situation in the brain. Possibly because TNF impacts endothelial cells, impacts oligodendrocytes directly. Interferon again, definitely is able to reduce the expression of the myelin protein gene, seeing that. So myelination is reduced throughout the brain. And then interleukin-8 is a typical cytokines driving an acute reaction, and it should not be present in the brain. And so, when we look at the history of our patient, we know that there is a progressive decrease in their cognitive functions. We associated it not with age, but with illness duration. Illness duration associates with reduction of fractional anisotropy throughout the brain, which leads to, mediates the effect on cognitive functions. A trial of trouble, that's how Ibrahim Aoun calls it. Abnormalities in glutamate activity, changes in astrocyte support of the neurons, activation of microglia and their impact on glia, on white matter, leads to a change, a big change in the homeostasis of the brain. It changes function, is a slowdown, and leads to both depression and cognitive dysfunction together. And they think that this is the pathway to the so-called neuroprogression, in which patients over the years have a worsening, progressive worsening of their cognitive function, which doesn't match that of healthy subjects. And this is achieved by impacting oligodendrocytes directly, changing all the corticolimbic circuitry, decreasing connectivity, and changing the level of the activity even at the level of synapses. Actually, we found, however, that there are a lot of cells in the brain coming from the periphery. This is the last thing I will say about the brain, and then we will see the drugs very quickly. Michael Schwartz clearly showed that through the choroid plexus, lymphocytes can get in the brain, and they go there in the presence of inflammation, of any kind of inflammation, in order to resolve it. If inflammation persists, if microglia continues to stay activated, the neuron will suffer and finally die, because the neuron needs astrocytes, support, metabolic support from all these cells, and when it lacks, we have the so-called neurodegenerative illnesses, such as Alzheimer's and so on. She studies in particular Alzheimer's. What lymphocytes do is to infiltrate the brain. They enter the parenchyma of the brain from the periphery, and they can resolve inflammation.
if an NK cell, a natural killer cell, is producing anti-inflammatory cytokines and find activated microglia, which is not responding to them, well, she is not so much diplomatic. The NK cells kills activated microglia. Boom, inflammation can be solved in one day, which is what happens during pathogenetic, let's say, the infectious diseases of the brain. So, what happens in the brain of our patients? Again, we don't know, but we think that this balance between the so-called protective autoimmunity, lymphocytes which enter the brain and target our cells in order to maintain homeostasis, and a lack of immunity is altered in immune disorders. This is a new way of thinking. Following this way, we showed that the peripheral levels of NK cytokine producing cells at the top of inflammation, at the top, I mean, of the control of immunity and inflammation, are linked with white matter integrity. Actually, the more cells, the more of these cells you have in the periphery, the better the white matter integrity in the brain of our patients. As if these cells would be part of the restoring of homeostasis, and they associate positively with fractional anisotropy and negatively with radial diffusivity, so with damage. Think that lithium acts on immunity and activates NK cells, and these mediate the effect that lithium has on white matter integrity. The more you are taking lithium, the better the white matter integrity in the brain, and this is partly mediated from a mathematical point of view by the increase in NK cells and in lymphocytes stimulated directly in the thymus. And we also show that other cells are linked with this. FOXP3 are linked with, which dampen immunity, are linked with worse white matter integrity, and TH17, which are cells for attacking, it's the adaptive immunity, defense against pathogens. The more the patients are having the periphery, and the better the white matter integrity throughout the brain. Second part of the talk, which will be very short, 5 minutes, I think, because I will try to run. I think when I start speaking about this, I never stop. You have to stop me, like they do with the from above, place somebody over there. So let's start with this. I will go very quick because this is a very well-known literature. There are a lot of papers clearly showing that the higher levels of inflammation in the periphery, the worse response to conventional antidepressants. This was done as soon as in the year 2000, 20 years ago. They showed that, you can see here, patients have higher levels of cytokines, of course, we have said it. But the non-responders at the baseline had really higher levels than controls and than the responders, which were not so different from controls. And then they all go down in this paper after treatment, and they were do not respond on the basis of these cytokine levels. So the explanation are not so easy. Yeah, this is the same stuff. This is again micromiles showing that higher levels of interleukin-6 and their receptors, you can see treatment-resistant depression has double levels in respect to treatment responding. And then here it is me showing, this is the last paper I published, it was January last year, showing that you can see here interleukin-1 beta. If you have low levels of interleukin-1 beta, or this is a 50% reduction, and these are the scores. Okay. If you have low levels of interleukin-1 beta, everything can happen. You can respond to treatment, non-respond to treatment. But at higher levels, patients tend to stratify toward a very bad response to treatment. Okay? And actually,
Yeah, well, no, I will say it again after the slide. Okay, so higher cytokines, low response. This is true also for these treatments, no pharmacological treatments such as C-depilation and C-physoldance. I didn't tell you that these people have been treated with both SSRIs, assorted SSRIs, or SNRIs upon clinical need. They are 120 patients, if I remember, in a common clinical setting. It's our patients as we treat them. And we showed that a panel of cytokines is especially predictive of non-response in bipolar depression. You can see here the levels of healthy subjects. White dots are patients who respond to treatment, and the black one are the patients who do not respond to treatment. But look here, this is not an absolute value, this is a fold change. This means 6 times higher than healthy controls. This means 4 times higher than healthy controls. Okay? So in bipolar patients, there are very high levels of activation who hamper response. And we also associated this with the effect of obesity and BMI, because we know that in yellow fat, there is an activation of macrophages which produce cytokines, and all the effect BMI can have on the predictor response to antidepressant treatment is mediated by this increase in cytokines. It's possibly positive feedback inhibiting response. The more I am inflamed, the more I will have metabolic problems, the more I will activate macrophages in my yellow fat, the more I will get again inflamed, and all this will have effects on white matter integrity and brain function and counter-antidepressant response to MCT1, which we associated with non-response. Okay? But then there is I contributed to it. This is a review I would suggest because it has to do both with this moving frame product I was speaking about, but also with the availability. And we can show it because here you have other treatment. You see citalopram, sertraline, venlafaxine, mipramine, and it links the non-response not only with the peripheral levels of cytokines, but with the expression of genes for producing cytokines in the circulating mediums. I'm telling also you that what we lack is a threshold in order to say above here we have no response, below we have response. This threshold has yet to be defined, but I think it's a good work for the future. Also because the ways of analyzing cytokines are so much different from one place to the other. And gene expression is the most reliable, but gene expression is also very much expensive and difficult to know. So, with immunophenotyping, it has not to be done. Okay? But I think in a future, again, with a small amount of blood from our patients, we could even choose drugs because we know that antidepressants share immunomodulatory and anti-inflammatory properties. Well, this has been said even during this COVID pandemic. It has even been proposed to repurpose these drugs because they target these endoplasmic reticulum resident protein sigma 1, which is a receptor with anti-inflammatory, let's say, cascades when stimulated. And we have a conventional antidepressant, you can see a lot of them, sertraline, fluoxetine, paroxetine, citalopram, escitalopram. All of them can target this receptor, and so they have an anti-inflammatory action, downregulate the production of interleukin 1 beta, 6, 8, and 12, which all have been associated with non-response. Okay? So one of the things that we do when we give antidepressants is to target inflammation. The point is that in certain papers there is a clear change. You see these guys published years ago, a high level of a lot of cytokines in patients related to controls. All of them are higher, and all of them decrease after 12 weeks of treatment, and the decrease
But when we do meta-analysis of the available data, and the last one, as far as I know, has been published 10 years ago, the evidence is not so striking. So probably we have a change in the long term, but not during an acute trials of antidepressants. You can see here, here there is some reduction in TNF, but overall it doesn't change so much. And looking at the single trials, these were 100 of people, and it didn't change, and a lot of them responded. This is venlafaxine at a very high level, and a lot of them responded, and the cytokines even increased. So we do not expect an immediate change in cytokines. Probably, this is again, who is this? Sorry. This is interleukin 6, buloxetine. Doesn't change, but people respond. What always is reduced is interleukin 1, probably because these drugs target the sigma receptor. But let's say that the evidence is not so striking. This matches with what we see into the brain. Let's go there again and look at the activation of microglia. You see that the most striking differences, these data come from the NIH, from the Unit of Experimental Treatment of Depression. You see that this is binding affinity in the subgenual prefrontal cortex and in the arteriocerebral cortex, and patients have higher activation of microglia, we know it. But when we look at the medication status, we see clearly that patients who are being treated with antidepressant drugs have a lower activation of microglia. So probably the effect of our antidepressants cannot be seen acutely in the changes in cytokines in the periphery, but needs to be seen in the long term, and this also matches with the possible benefit that they give to the patients in terms of brain integrity. This is, I think, a beautiful study. These are the years of untreated major depression, and this is the binding of the SPO in untreated major depressed patients in the insula, interterminal in the prefrontal cortex. The more time I stay without treatment in depression, and the worse the activation of microglia into my brain, and the worse the structure and the function of the brain. This needs to be studied now. My little studied this acute chronotherapeutic intervention with sleep deprivation and light therapy, and I showed, according to what Raymond Land had shown many years ago in one of the first slides of my talk, that in respect to healthy controls, some of the patients have very high levels of interleukin 1 beta in respect to its receptor antagonist, and a successful decrease of this marker of inflammation do associate with the response to treatment, but there are sparse evidence. Finally, I will take 3 minutes, okay? Finally, we can use drugs which target the immune system in order to obtain antidepressant effects. And this, I think, is a completely new field for the future. This is celecoxib, a COX-2 inhibitor. When added to reboxetine, it leads to a better effect. There are not so many data about this. This is again celecoxib, but you can see it's not such a big difference over time. So why should we give these drugs full of side effects, Well, probably what we should do, and again, there is a striking, I think, finding about this, is to give it to the patient who have a very high level of baseline inflammation. Overall, the difference between adding celecoxib or adding placebo is not so big, but it is proportional to the change in serum interleukin 6 as a marker of inflammation obtained in patients who are being treated, and this change is proportional to the baseline level, as it is shown by another study.
here we are, which comes again from Savitz and Brevetz and people of the NIH, you can see that the responders treated with, in this case, minocycline, which is another individual of, it's a tetracycline which can cross the blood-brain barrier and inhibits microglia, you can see that the responders are the people who had high levels of interleukin-6 at the baseline. So if you don't have this inflammation, adding an anti-inflammatory drug will not give you a particular benefit. The data tells us that we should possibly in the future stratify the patients based on the basal level of inflammation and then treat them with a personalized individual treatment to target. Let's go again into the brain. This has been published on biological psychiatry now. I think it's out. It is like time ago. You can see that at the low levels of TSPO, patients treated with celecoxib do not have any improvement. But the patients who have the highest level of activated microglia are the patients who have the best benefits from adding a COX-2 inhibitor. And there is a non-linear relationship between the two. It's very nice the finding, in this sense, the very famous finding by Andrew Miller, by Rayson, with infliximab. Infliximab is a blocker of TNF, but careful, because one says, okay, TNF alpha, inflammation. Yes, when it binds to the soluble receptor for TNF alpha prompts inflammation. But when it directly binds to cells, it even has anti-inflammatory and neurotrophic properties. So what do? We block TNF, and what do we obtain? Nothing. This is an ADAPT study in resistant depression to drugs, and you can see infliximab or placebo, and this is placebo. It is slightly better. What did they do then? They disentangled, they stratified the patients based on the level of C-reactive protein at the baseline. If C-reactive protein was low, the placebo wins over infliximab because probably infliximab blocks these effects of TNF which are neurotropic. But when CRP is high and the patients do not respond to treatment, at that point, blocking TNF and blocking inflammation is useful. So it's not for everybody, okay? It's a targeted treatment, this is the idea, for patients who have an inflammatory reaction. And look here, scores in the childhood trauma questionnaire, physical abuse associated with clinical severity, C-reactive protein, and response to infliximab. The patients with worse inflammation, whatever the cause, even childhood adversities, it has been published on JAMA Psychiatry just before the COVID, do have benefits. You see, if they were overall, infliximab does not give any help. And in these childhood abused adult patients, infliximab was much superior to placebo in targeting their persistent inflammation in the brain. And then we now have this minocycline, which is an adjunctive treatment. Again, there are several studies, single studies showing that it is superior to placebo, sometimes with great effects. I'm not so sure of those. What I trust a lot is this study because they, again, this is the group of Camille Paviante, Benedetti, and the King's College. And they showed that you see, if the patients had high baseline levels of C-reactive proteins, they responded and they decreased their C-reactive protein and Hamilton 17 score. But if they had low levels of C-reactive protein, the decrease in Hamilton ratings of impression was not significant. And so, these are the take-home messages, at least at the end. Sorry for stealing 10 minutes. It was 1 hour I took, we started with 5 minutes late. I think it was good, it's staying here. We don't have time for discussion, but I will be here until the evening, so you can ask questions whenever. So I showed you that patients with good disorder clearly have something big impacting their immune inflammatory system. We don't know it so well. I think one of the reasons, there are two main reasons. First, the phenotype is not stable over the life. It worsens progressively. So, if you look at the literature, they mix up patients with different ages, but probably we have different phenotypes in the lifetime of the patients, and these are individual changes over time. Also linked with the functioning of the biological clock. And the effects on the brain, on the contrary, are very, very clear. Activation of microglia, disruption of white matter integrity. I didn't show the data about functional connectivity. I will show them to you this afternoon in the post-COVID depression. But of course, disrupting white matter connectivity does impact functional connectivity of the brain, functions, cognitive functions, depression. The higher the inflammation, the worse the depression. Even suicide can be a consequence, and cognitive deterioration and neuroregression. Anyway, it's very difficult to study them because at this point we do not have an MRI setting for individual patients. We can't do, because all what we can see even at 3 Tesla is normally appearing white matter, and just with these quantitative measures we can find these differences and these effects. But in the future, I hope we will be able to scan our patients and have a clinical picture for this. And then, all these effects of the drugs with the basic idea that worse inflammation is also a problem for response to conventional antidepressants, and these cases, not all the cases, but these cases of very high inflammation will be selected for specific anti-inflammatory and immunomodulatory treatments to be used as antidepressant in the clinical practice. That's it. Thanks for listening. Time must go. I don't know if